Welcome to General Microbiology, a channel that provides a high-level overview of everything you need to know about bacteria, viruses, and other microorganisms. Today we'll be talking about bacteria, its shape and structure, as well as function of its general components. Bacteria are a type of biological cell and comprise a large domain of prokaryotes together with archaea. Although it is important to note that other models of the tree of life have been proposed and are still relatively actively debated. Bacteria are typically considered unicellular, although recent studies have shown that this concept doesn't fully explain bacterial behavior in colonies and biofilms. They take on an array of shapes and sizes, ranging from spheres to rods and spirals. Bacteria inhabit nearly every place on Earth, from radioactive waste to acid hot springs and the polar ice caps. By mass, bacteria are estimated to exceed both plants and animals, with organisms in excess of 5 times 10 to the power of 30 on the planet Earth. That is just an estimate, but nevertheless very impressive and thought to be outnumbered only by viruses, the most predominant of which, not very surprisingly, infect only bacteria. When it comes to size, bacteria are generally 10 to 100 times smaller than eukaryotes. As an example, a macrophage, one of the most critical cells of our immune system, is about 10 micrometers in diameter, whereas a spherical bacteria, Staphylococcus aureus, is about 1 micrometer in diameter. They're about 10 times larger than most viruses. As an example, an HIV virus particle is about 120 nanometers or 0.12 micrometers in diameter, about 10 times smaller than that Staphylococcus aureus, which was about 1 micrometer in diameter. The spherical bacteria are generally called cocci. Rod-shaped organisms are termed bacilli or coccobacilli. However, a whole array of other shapes have been defined, including vibrio, helical forms such as the famous Helicobacter pylori, spirochete, and filamentous. The outermost structure of the bacteria is the capsule. Not all bacteria produce capsules, and those that do can produce it transiently, turning the production on and off depending on the surrounding environment. The capsule can help the bacteria attach to surfaces, prevents phagocytosis and destruction by immune cells, limits the diffusion of molecules and viruses that could be damaging to the organisms, and can even serve as a mechanism to prevent bacterial dehydration. Some capsules are comprised of polysaccharides, while others of proteoglycans and glycoproteins. Some are densely packed around the organism, and others tend to be larger and blend in with the environment. Some bacteria also have a flagellum, a whip-like organelle that helps bacteria movement and sensing. Flagellum is present on some eukaryotes, and a similar structure, albeit under a different name, can be found in archaea. The flagellum is a multi-protein organelle, predominantly comprised of the protein flagellin. This bacterial motor can turn up to 1,000 RPM, is able to change direction, and is powered by a flow of protons and sometimes sodium ions. The bacteria can have one flagellum, 2 on each end, or multiple flagella on various ends of the organism. Many gram-negative and some gram-positive bacteria also possess smaller and shorter appendages called the fimbria. These are used for attachment and adhesion. Fimbria contain adhesins, which are proteins that are able to attach with relative specificity and withstand substantial shear force. Some bacteria use fimbria to obtain nutrients and move. However, this organelle is predominantly used for attachment and adhesion. An appendage whose classification is at times debated is the pilus. It is fewer in numbers than the fimbria, but projects beyond the range of a typical bacterial fimbrial appendage. The main purpose of the bacterial pili, or singular pilus, is for conjugation, or exchange of genetic material between the bacterial donor and the recipient. The pilus, like many external appendages, is antigenic, and can be readily replaced by the organism and is only expressed in certain environmental situations. The bacterial cell wall greatly varies between organisms and is typically subdivided into gram-positive staining and gram-negative staining. Both types of organisms have an inner membrane comprised of a bilayer of fatty acids, a host of proteins embedded into this membrane, and an outer paramembrane space right above it. Both gram-positive and gram-negative bacteria have peptidoglycan, a polymer of sugar and amino acids. However, gram-positive organisms have significantly more peptidoglycan in their cell wall. In addition, teichoic and lipoteichoic acid molecules are intertwined within the outer peptidoglycan layer of gram-positive organisms. Gram-negative organisms, on the other hand, have an outer membrane, which is another phospholipid bilayer with proteins embedded in it and lipopolysaccharides hanging off the outside. These lipopolysaccharides, also termed lipoglycans or endotoxins, are immunogenic and toxic. Gram-negative bacteria are able to secrete these into the environment, and if released into the human body, these molecules can cause fever, diarrhea, and possible fatal endotoxic shock, also termed septic shock. The gram stain is a very important staining method in microbiology, which uses dyes to color bacteria prior to visualization. Gram-positive bacteria owe their purple color retaining ability to the thick layer of peptidoglycan that comprises 50 to 90% of the cell envelope. Despite having peptidoglycan, the gram-negative cells are unable to retain the gram stain since the peptidoglycan layer is typically very thin, comprising only about 10% of the envelope. As a result, the gram-negative organisms appear pink to light red on the gram stain protocol. While the internal structures of a bacteria are not as complex as those of most eukaryotic cells, they are still worthy of their own overview. At a high level, on the inside of the cell membrane lays an array of nutrients, proteins, lipids, polysaccharides, RNA, and DNA. Unlike eukaryotic cells, bacteria typically lack large membrane-bound organelles. However, protein-bound compartments, such as the carboxysome or lipid-enclosed chlorosomes, also termed mesosomes, are at times present and serve various functions. Instead of the nucleus, bacteria have a nucleoid, with typically a single circular bacterial chromosome. Bacteria also house an array of extrachromosomal vectors, many of which can code some of the most potent virulence factors. In addition to all of this, bacteria contain ribosomes, which are the molecular machines found in all living cells and responsible for protein synthesis. And of course, the interior of the bacteria are full of intracellular structural components, proteins, enzymes, substrates, and RNA, among many other highly specialized molecules. Each of these, their function, structure, chemistry, and relationships amongst each other are going to be the topic of other videos. For now, thank you for tuning in and hope you enjoyed this brief overview of bacteria.